Hello everyone, welcome back to our channel Simple Bio Lectures, and today we are discussing on the topic types of tissue culture. So yeah, basically there are three types of tissue cultures. First is the organ culture, second is the explant culture, and the last is the cell culture. So we will talk in detail about each of these three tissue cultures in the upcoming slides. So yeah, first we start with the organ culture. So organ culture, as the name suggests, it implies that the architectural characteristic of the tissue in vivo is retained, at least in part, in the culture. So in organ culture we use a complete organ as for tissue culturing, and towards this end the tissue is cultured at the liquid gas interface, which favors the retention of spherical shape or the three-dimensional shape. So as you can see in this figure, this is our organ, this is the liquid, and this is the gas. So our organ is placed at the liquid gas interface, and because of this the three-dimensional shape of the organ is maintained. Next we move on to the explant culture. So in primary explant culture, a fragment of tissue is placed at a glass liquid interface, whereafter migration is promoted in the plane of solid substrate. So yeah, in organ culture we used a complete organ as a tissue culture. In explant culture, what we use is a fragment of a tissue, not the complete organ, but the fragment of that organ. So how we will culture is, we will culture it on the glass or plastic and liquid interface. So this is our fragment of the tissue, and when we zoom in, we can see that it will grow, the cells will grow on the solid substrate. So on both these directions cell will grow. Okay. Next we move on to the cell culture. Now cell culture implies that the tissue or outgrowth from the primary explant is dispersed either mechanically or enzymatically into a cell suspension, which may be then cultured as an adherent monolayer on a solid substrate or a suspension in culture medium. Yeah, so it seems very difficult, but it is actually very easy. So in organ culture we use the complete organ as a tissue culture. In explant culture we use a fragment of tissue. In cell culture we use just the cells, which are like derived from the primary explant, either by mechanically or enzymatically dispersing it. Okay, so cell culture is just the cells which we use for tissue culture, but there are two types. So first we can use it as adherent monolayer on a solid substrate. So yeah, you can see on this, this is the single monolayer which is formed on the solid substrate of like individual cells, or we can use in a suspension in a culture medium. So suspension is just like media in which the cells are suspended freely. So the cells will roam around freely, there is no substrate to stick on to. So these adherent monolayer cells are known as anchorage dependent cells, and the cells which are in suspension are known as anchorage independent cells. So yeah, that's it for today. Thank you for watching this video. Please subscribe our channel.